Assalamu alaikum and welcome everyone to our molecular biology ASCP preparation series. This program is all about providing real-life MCQs for your certification success. The MCQs we'll discuss today and in the upcoming session have been carefully collected from several years of past exams to give you the best preparation possible. I'm Dr. Raya Abdelati, and I'm really excited to share my knowledge and experiences with all of you through this series. I also encourage everyone taking the test to share your question with us so that everyone can benefit. So let's dive in together, but don't forget to share, like, and subscribe to our channel to receive all the latest lecture. The molecular science part is divided into four sections: nucleic acid chemistry, basic molecular theory, biochemical reagents, and genetics, which together make up 20 to 25% of the total molecular biology ASCP exam. In our previous lectures, we completed the multiple choice questions related to the nucleic acid chemistry in lecture 1 and 2. We then moved on to the basic molecular theory section, beginning with the replication process. In the lectures 3 and 4, we covered DNA replication in eukaryotics. Now we'll focus on the replication process in prokaryotics. All right, so let's move on. All right, now let's focus on the question related to the DNA replication in prokaryotic and review molecular biology ACP questions from the previous years. All right. All right, the first question we have it today is: which prokaryotic DNA polymerase is primarily responsible for lagging strand synthesis during DNA replication? All right, in this question, we encounter different types of DNA polymerases, and I will explain their function to help you remember them. All right, this is important because similar questions are frequently repeated in the exams. So let's break it down. Actually, the correct answer here is DNA polymerase 3. DNA polymerase 3 is the primary enzyme responsible for sensitizing new DNA on both leading and lagging strands. Alright, this is the first point. What about DNA polymerase 1, 2, and 4? For DNA polymerase 1, removes RNA primers from the lagging strands Okazaki fragments and replaces them with DNA. Alright? For DNA polymerase 2, play a role in DNA repair process. DNA polymerase 2 play a role in DNA repair processes. For DNA polymerase 4, is involved in translesion synthesis, allowing for what? Allowing replication to bypass damaged DNA. Okay, so that understanding this distinction will make it easier to answer exam question on these topics. Actually, no need at all to worry about that memorizing all these details of the DNA polymerases enzyme right now. I have already arranged it in the table at the end of this slide for you, so you can review later. All right. So now let's move to the next question. Okay, the next question is: which prokaryotic DNA polymerase is primarily involved in DNA repair mechanism and replication start at next and lesion? All right. All right. The correct answer is DNA polymerase 2. As I said, DNA polymerase 2 is involved in DNA repair processes, particularly in the context of repairing damaged DNA. All right. Let's move on. All right. Which prokaryotic DNA polymerase is primarily responsible for sensitizing the leading strand during DNA replication? correct answer is DNA polymerase III. DNA polymerase III is the main replicative enzyme in prokaryotes, alright? Responsible for sensitizing the leading strand during DNA replication. It sequentially adds nucleotides to the growing DNA chain, forming a continuous complementary strand to the template DNA. Alright? So the correct answer is DNA polymerase III. Okay, the next question is which DNA polymerase is primarily responsible for the elongation of the RNA primer during lagging strand DNA replication in prokaryotic? Alright? Alright, the correct answer is DNA polymerase III. But let me walk you through the steps involved in lagging strand DNA replication. It begins with the priming, where primase sensitize a short RNA primers on the lagging strand. Alright? Following this, during elongation, when DNA polymerase III extend the RNA primers by adding deoxyribonucleotides. As the DNA polymerase III reach the end of one RNA primer, what will happen? It dissociates from the template, allowing primase to sensitize a new RNA primer for further elongation. And finally, during ligation, when the RNase H remove the RNA primers and DNA polymerase I fill the gap with the DNA nucleotides. Alright? Then ligase seal the next between adjacent Okazaki fragments and completing the process. Alright? So the correct answer, as I said, is DNA polymerase III, as it is the primary enzyme responsible for adding nucleotide to the growing DNA strand or lagging strand. Alright? Let's move on. The question here is the strand on which DNA replication is continuous is called the correct answer is leading strand. As we know, leading strand is sensitized in the 5' to 3' direction following the replication fork as it unwinds. Alright? In contrast, the lagging strand is sensitized discontinuously in a short fragment called what? Called Okazaki fragments, which are later joins. Okay, let's move on. Which enzyme initiates DNA replication in prokaryotes by unwinding the DNA helix? The correct answer is helicase. Helicase is responsible for unwinding the double-strand DNA at the replication fork, creating two single strands that serve as the template for replication. All right, let's move on. Okay, let's see the next question. What is the function of DNA polymerase III in prokaryote DNA replication? Actually, DNA polymerase III, as I said, is the main enzyme responsible for sensitizing new DNA strands in prokaryotic replication. It adds nucleotide to the RNA primer, alright? Extending the leading strand continuously and lagging strand discontinuously as Okazaki fragments. Alright? Let's move on. Which protein prevents the reannealing of single-strand DNA during replication? The protein that prevent the re-annealing of single-strand DNA during replication is called single-strand binding protein. But how it works? The single-strand binding protein binds to the single-strand DNA exposed during replication and stabilizes it, preventing the strands from re-annealing or forming secondary structures. This ensures that the DNA remain available as template for replication process. All right, let's move on. What is the role of DNA ligase in prokaryotic DNA replication? Actually, DNA ligase seal the necks between the Okazaki fragments on the lagging strand, forming a continuous DNA strand. So the correct answer is A, joining Okazaki fragments. All right, let's move on. All right, during prokaryotic DNA replication, which enzyme removes the RNA primer and fills the gap with the DNA? All right, the correct answer is DNA polymerase 1. DNA polymerase 1 removes the RNA primers and replaces them with what? With DNA nucleotides. All right, let's move and see the next question. Okay, the next question is which enzyme relieves the supercoiling tension ahead of replication fork in prokaryotes? All right, the correct answer is topoisomerase. Topoisomerase relieves the supercoiling tension by making temporary cuts in the DNA and then resealing it. All right, let's move on and see the next question. Okay, which part of prokaryotic DNA replication process ensure high fidelity of DNA replication? The correct answer is proofreading activity of DNA polymerase III. This enzyme possesses a 3' to 5' exonuclease activity, allowing it to remove incorrectly paired nucleotides and replace them with the correct ones during DNA replication, ensuring high fidelity. All right, let's move on to the next question. What is the role of DNA gyrase in prokaryotic DNA replication? Actually, DNA gyrase is the type of topoisomerase relieves the supercoiling tension that form ahead of the replication fork during DNA replication. So the correct answer is relieving supercoiling ahead of replication fork. All right, let's move on. Which enzyme seal the necks between the Okazaki fragments on the lagging strand? The correct answer is DNA ligase. DNA ligase seal the necks between the Okazaki fragments, forming a continuous DNA strand on lagging strand. All right, let's move on. Okay, the next question is what is the primary function of the beta clamp in prokaryotic DNA replication? Actually, the beta clamp ensures that the DNA polymerase remains attached to the DNA. allowing it replicate the DNA efficiently and processively. All right. So the correct answer here is ensuring the DNA polymerase remains attached to the DNA. All right. Let's move on. The next question is, which protein complex recognizes the origin of replication in prokaryotes and helps to initiate the replication process? The correct answer is DnaA. DnaA is the protein complex that recognizes the origin of replication in prokaryotes and helps to initiate the replication process by unwinding the DNA at the origin. All right? Okay, the next question is, in prokaryotic cells, which enzyme sensitizes the leading strand continuously? The correct answer is DNA polymerase III. DNA polymerase III sensitizes the leading strand continuously in the 5' to 3' direction during DNA replication in prokaryotic cells. Alright, let's move on. Alright, which protein helps to prevent the single strand DNA from forming secondary structure during replication? Alright, the correct answer is single strand binding protein. Actually, single strand binding proteins bind to the single strand DNA to prevent it from forming what? Forming secondary structure and re-annealing during replication. Alright, let's move on. The next question is, during prokaryotic DNA replication, what is the function of primase enzyme? Actually, Primase sensitizes short RNA primers that provide a starting point for DNA polymerase to begin DNA synthesis. All right. So the correct answer here is sensitizing RNA primers. All right. Let's move on. All right. The next question is, which enzyme is responsible for proofreading newly sensitized DNA in prokaryotes? The correct answer is DNA polymerase III. As I said earlier, DNA polymerase III has proofreading activity that ensures high fidelity by correcting any errors during DNA replication. Alright, let's move on. Here we see the next question. What is the role of DnaB protein in prokaryotic DNA replication? Alright, the correct answer is unwinding the DNA double helix. Actually, DnaP, which function as the helicase, unwinds the DNA double helix at the replication fork, allowing the replication machinery to access the DNA strands. All right, this is the final question we have it today. Thank you for your attention. And in the next slide, I will provide overview of key prokaryotic DNA polymerases. All right. All right. Here is the overview of key prokaryotic DNA polymerase 1, 2, 3, 4, and 5. Be sure to review and remember this information as it frequent focus in the exam, right? Pay particular attention to the DNA polymerase 1, 2, and 3 they are most important one. All right. Yes, here the 4 DNA polymerase 4 and 5, but you need to pay attention to the 1, 2, and 3. All right, but you can pass through this one. All right, that's all for today. Please don't forget to comment below if you have any questions. Share, like, and subscribe to our channel to receive all the latest lecture. Salam.